Okay, so in this video, I'm going to go over chapter 5, which talks about environmental influences and control of microbial growth. So we're basically now going to go into how microbes grow and the different conditions that they require to grow. And obviously, different types of bacteria have different growth requirements, but we're going to talk about kind of the different things that influence how they grow. Okay, so this is the outline. So we're going to talk about first the pressure and temperature, how those kind of limit the growth and how certain bacteria like high temperature, high pressure, things like that. Then we'll go over osmolarity. So this has to do with salinity primarily. And then we'll do pH and oxygen levels. So again, this is all just talking about what certain conditions do these bacteria like to grow in. Then we're going to go over nutrient deprivation. So now we're going to talk about what goes wrong, what happens when there's low amounts of nutrients. How do these bacteria kind of react to that? And then finally, we'll go over controlling microorganisms. And that means us as humans. How do we control them and prevent them from growing? So we'll talk about different ways to do that. Okay, cool. So this is a table that kind of just does a quick overview of everything we're going to talk about when it comes to their environmental parameters or their environmental conditions. So like I said, there's temperature. So different types of bacteria grow at different temperatures. We have pH, so there's different pHs. Osmolarity, again, usually has to do with the salinity or salt concentration. Then we have the oxygen levels and pressure. So we're going to cover all of these in different slides. Okay. So let's first talk about temperature. So different bacteria have different temperature preferences and their growth rate changes at different temperatures. So the psychrophiles are going to be the ones that grow in very low temperatures. So as you can see, they like it pretty cold. I mean, 0 degrees Celsius is basically freezing temperature. They don't prefer it, but they could maybe grow close to freezing. They're optimal. So the peaks is basically showing the optimal temperature. That's where their generations per hour or their growth rate is the highest. So these psychrophiles typically like 15 degrees Celsius. So I'm trying to think what that's cold. It's not as cold as a refrigerator. So refrigerators are anywhere between 4 to 8 degrees Celsius. So that would be over here. So these guys could still grow in the refrigerator, but not really well. They prefer a little bit warmer. Keep in mind, room temperature is anywhere between 20 to 25. So it's colder than room temperature, but not as cold as the refrigerator. So whatever that temperature is. Okay, so those are the psychrophiles. Then we have the mesophiles or the mesophiles. They prefer around 30 to 40 is their best temperature. So I call them the normal ones. They're the ones that grow at normal temperature. They grow at room temperature. So 25 is around here. And they prefer the best is body temperature is their preference. So as you can see, their peak is around 37 degrees Celsius, which is human body temperature. So that's their ideal. Okay. Then we have the thermophiles. So these guys, obviously, as you can tell, they prefer very hot temperatures. So anywhere between 50 to 60 degrees Celsius. That's pretty hot. I mean, it's not boiling. Boiling temperature is at 100, but that's still pretty hot. It's definitely hotter than the human body, twice as hot, actually. So, and then finally, we have the extreme thermophiles, which guess what? They really like it hot. So some of them can even survive in, boiling temperatures. So those are the ones that you're going to see in like the hot springs where there's bacteria growing in like those hot springs outside. And so they like it super hot. And so those are those guys. And notice that they actually not only like it super hot, but they couldn't grow in body temperature. So for example, if I got an extreme thermophile from a hot spring and tried to infect myself with it, it wouldn't grow in my body. So also keep in mind that their range also limits them. So for example, a thermophile grows really well at hot temperatures, but again, would not be able to grow at body temperature, which is around 37. So again, they have their limits. They are able to grow at really high temperatures, but not at low. And same for the psychrophiles. So they can grow at really low temperatures, but not hotter than that. So they have their limits. Okay. So that was temperature. Now we're going to go over pressure. So pressure, why would a bacteria even be exposed to different pressures? Well, some bacteria live at the bottom of the ocean, and some bacteria grow at the very top of a mountain. And so those are very different pressure, locations. So the, regular land at sea level where we are living, the pressure is close to 1 atm, which is the standard pressure. Whereas as you go deeper into the ocean, the ATM gets higher. I mean, at the very bottom of the Mariana Trench, it's like 1,000 ATM. That's 1,000 times higher pressure than what we're currently living in. So that's like super high, right? Whereas at the top of Mount Everest, it's going to be almost a third of the pressure that we are currently in. And so obviously certain bacteria have adapted to these different pressure ranges. Okay. I'm not really sure why this graph has a pressure in megapascals, because I don't really have it mentally. I don't really have an idea what the megapascal is. Like, I'm used to ATM, but whatever. Here is the megapascal graph. So we have the barosensitive. So down here are these organisms called barosensitive. That means that they cannot live in high pressure. So notice that they prefer low pressure. And then as you increase the pressure, it gets crazier and crazier. So actually, it's starting to bother me. I need to figure out the conversion between megapascals and ATM, because it just doesn't make sense. I'm just going to do it on my phone really quick. Okay, so if we did 20 megapascals, oh my God, that's really high. Okay, it's like 200. That's crazy. Okay, so here, let me just write in here. So 20 megapascals is approximately, it's not exact, but it's approximately 200 ATM, which means that this is approximately 400 ATM at the 40 mark, and so on. And so this right here is at approximately 1,000 ATM. Again, approximately, right? The conversion is not perfect, but that's just what I'm estimating, according to Google. Okay, so that's like really high. Okay, but that gives me a better idea of what we're working with. Okay, so anyway, the barosensitive, they really don't like it where the pressure is high, right? They like it almost near zero, not totally zero. So let me see what 1 ATM is in megapascals. Okay, so just to give you an idea, 1 ATM, which is the normal pressure that we are currently living in, is the same as 0.1 megapascals. So that gives you an idea that these organisms here like to live in pretty much normal pressure. Okay. Anyway, so those are the barosensitives. The barotolerants are these organisms that can grow at high pressure. They can also grow at low pressure, though. So they can grow at low pressure, high pressure. They can do it all. They do have a limit, though. They can't go anywhere past like 60-ish. They start to die out past 60 megapascals. So that's 600 ATM-ish. Okay, and then we have the barophiles. So the barophiles are going to be shown in red in this graph. And just kidding, not red, blue. The barophiles are in blue in this graph, and they only can grow at high pressure. So that's the difference between the tolerance and the phials. So phials typically means that they have to have that condition to be able to grow. So notice that the barophiles are not able to grow at low pressure, and obviously not at super high pressure, but they have an optimal pressure, and that is at around, I guess, 550 megapascals. So again, with the barophiles, whenever you see the word phial, it means that they have to have that kind of condition. Whereas the tolerant just means, okay, I can tolerate it. I can grow in that condition, but really, I probably don't prefer to be living in high pressure, but I can if I have to. Okay. And then, yeah, again, make sure that you realize that there's not really too many organisms that can grow beyond 80 to 100 megapascals. Again, that's 800 to 1,000 ATM approximately. And I say that as we know right now, we don't really know that many organisms that can live in these high pressures. Now, we are obviously doing research at the Mariana Trench, so I'm sure that people have found some organisms that are growing at that super high temperature. At that super high pressure, but at the moment, we don't really have any organisms that do, or at least to my knowledge, that can go that high in pressure. Okay, cool. All right, so now we're in 5.2, and we're talking about osmotic pressure. And before we talk about osmotic pressure, let's talk about aquaporins. So aquaporins are these proteins, membrane proteins, that allow for water to pass through the cell membrane. Remember that the cell membrane is selectively permeable, and water is hydrophilic because it is water, right? It's not even hydrophilic, it's just water. And so water does not pass through the cell membrane very well because the cell membrane is hydrophobic. So these aquaporins are very important for bacteria if they are put in a situation where there's osmotic stress. So osmotic stress means that there's either a very high salt concentration or low salt concentration. So you're going to have to review back to your osmolarity and like osmosis in general biology, where, there's like the hypotonic versus hypertonic versus isotonic. So if you're like, what is that? You might want to pause and just quickly Google search what that is. But anyway, so these aquaporins help regulate the amount of water that's going in and out of the cell. When it's either hypertonic or hypotonic, whatever. So super important. So yeah, regulates the osmotic pressure. Another way that bacteria can regulate themselves if they're put in a situation that's super salty, for example, is these compatible solutes. So what a compatible solute is, it's a molecule that will not interrupt the cell if there is a very, very high concentration inside. So most molecules will cause some kind of disruption when there's way too much. So for example, if a cell has like an extreme amount of sodium inside, that cell's probably not going to live. So that's what we call an incompatible solute. But compatible solutes are molecules where even if you had like a crazy amount on the inside of the cell's metabolism and lifestyle doesn't get interrupted. And so those are what we call compatible solutes, because what these bacteria do is they purposefully take in these solutes to balance out the osmotic pressure from the outside. And so this usually happens when the cell is put in a situation where it's hypertonic. So hypertonic means that there's so much solute on the outside. So here's your little bacteria. And let's say that there's like, a lot of sodium on the outside. Okay, so this is a hypertonic solution. Now, if you remember from Gen Bio, what happens when a cell is put in a hypertonic solution? What happens to the water? And actually, go ahead and pause the video and think about where the water is going to move. Is the water going to move out of the cell or into the cell in this situation? Again, this is hypertonic. Okay, so what's going to happen is that the water will be drawn out, right? So water will leave the cell. That's bad, obviously. The cell needs water. And so the cell will shrivel up and die, right? So that's not good. And so the way that a cell, bacterial cell, kind of alleviates this is they start to produce a bunch of these compatible solutes. So these compatible solutes are really small molecules. I think I can't remember off the top of my head, but there's like a few like amino acids and some sugars that they can produce. So I'm just going to put CS for compatible solute because there's like a bunch of different types, but CS, 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 CS. So if there's a bunch of compatible solutes inside, then the solute concentration outside and inside are balanced. And so now we've created, quote unquote, an isotonic solution. So then water goes back in, and now it's isotonic. Now, it might not be perfectly isotonic, but at least it alleviates the situation. And so now some of the water goes back in. Okay, so lots of sodium on the outside, water leaves the cell is now freaking out because it's losing water. So it starts to produce these compatible solutes, which draw the water back in because now it's creating a hypertonic solution on the inside. And so then the water is drawn back in. And because they're compatible solutes, meaning that they don't really interrupt the cell when the concentrations are really high, then the bacteria live. So this is an adaptation that organisms like halophiles can do. So a halophile is an organism that really likes salt. So this is a picture of a salt lake, like a really, I guess it's pink because of the salt. Oh, no, it's pink because of the bacteria, but it's a very high concentration of salt. Okay. So before we talk about the halophiles, let's talk about the halotolerance. So tolerant, again, remember, it means that the organism can grow in that salt concentration. It tolerates it, but it definitely probably doesn't prefer it, but it can do it if it needs to. Now, the halophile, remember the word phile means that the organism can only grow in that condition. So in this case, organisms can only grow when there's a high salt concentration. And actually, these halophiles require really high salt, 10 to 20% salt, which is like really crazy. Think about the human body. Normal saline is like what, 0.8%? I can't remember. 0.85 is normal saline, which is like the salt concentration of the body. Let me check. concentration 0.9 or 0.85. Yeah, so 0.9%. That's less than 1%. So these halophiles are living in conditions that are 10 to 20 times more salty than anything that we've ever been familiar with. So that's really insane. So yeah. Okay. All right. So now let's move on to 5.3, which goes over pH and growth. So again, different types of pHs, different organisms can tolerate different pHs. So this one's pretty easy. So neutralophiles, guess what? Grow in neutral pH. Acidophiles, acidic pH, and alkalophiles in basic pH. So that's pretty easy to remember. Okay. Something to keep in mind though is that these organisms can live in this pH, but it's not because their internal or not organs, their internal self has that pH. They are just able to tolerate the pH that's around them. So if it's like super acidic on the outside, it doesn't mean that it's super acidic on the inside of the cell. The cell is able to regulate its own internal pH regardless of what's going on the outside. So these acidophiles are able to basically grow in a very acidic pH, but still maintain an internal neutral pH. Same for the alkalophiles. And so let's talk about one mechanism in which these organisms kind of maintain their own pH. And it's a little bit weird. So it's called the sodium plus or sodium ion circulation, and it helps specifically when there is a basic pH stress. So when an organism is living in, so I guess an alkalophile. So if you have an alkalophile and it's living in basic pH, it does these things to prevent itself from being overstressed. So let's first review. So a basic pH means that it's a pH that is higher than 7, which means that the concentration of the H plus ions is very low, right? Acids have high H plus and bases have low H plus. So right now we're talking about a situation where there's not a lot of H plus, which is problematic. Now the biggest problem is, and you might remember this from a previous chapter, is that hydrogens are used all the time in the cell for proton motive force. Remember that proton motive force is what powers ATP synthase. It's also what powers the flagella. There's like a lot of things that use it. And so one of the biggest issues that these organisms face is that the pH is too high. And so the Na, the H plus concentration is too low. And so they cannot produce this proton motive force. So what they've done is adapted and produced an Na plus motive force, meaning that instead of protons moving, it's sodiums that move to power certain things. So for example, their flagella is modified so that instead it uses sodium motive force. So the movement of sodium inside the cell, the movement of sodium into the cell is what powers the flagella. So that's just an example. Another example over here is like this Symport molecule is also using sodium that's going inside. Okay, great. So as you can see, all of these things are causing sodium to go inside of the cell. So now the cell has a very high sodium concentration, which is going to be problematic. And so now the second issue, so the first issue was, shoot, we don't have enough H plus, so we need to figure out another way to power our reactions. And so the cell decided, instead of using H plus as my motive force, I'm going to use, so instead of proton motive force, I'm going to use Na plus motive force. Okay, that's great. And so that's what's happening here in the bottom of the diagram. Okay, but then that causes a problem because now it's super high concentration of Na inside, right? And that's also problematic. So then it also has adapted ways to get rid of that Na. So one way is using the NaH plus antiporters. And remember, an antiporter means that one goes in and the other one goes out, right? And so the sodium leaves the cell, okay? And this is going from high to low concentration. So this one is the one that is happening through, diffusion, essentially. And that energy is going to power the movement of H plus into the cell, okay? So actually, this solves two problems. You're getting rid of this excess Na, and you're actually recruiting H plus, which is really good because remember in the very beginning, we were talking about the fact that there wasn't enough H plus in the system. So bringing that H plus in is really good, right? The cell's like, oh yeah, hell yeah. So this actually is like kills two birds in one stone. You get to get rid of your excess Na plus and acquire more H plus, which is very low right now in this environment. Okay. And then now that we have acquired some H plus, the cell will pump it back out, which is ridiculous to think about, but the H plus will get pumped right out. Okay. And so we call that the respiratory chain or respiratory pump. I don't know why yeah, so respiratory chain pump, whatever. It's like a very long name. But anyway, so the H plus just leaves the cell through this respiratory chain. Okay. And that is pushed out. And then guess what? It comes right back in. So what the heck is the cell doing? It's just pumping H plus out and then bringing it back in. It's kind of weird, right? Um, but anyway, what happens is as it's pumping all of these hydrogens out, it produces a concentration gradient, which then brings the hydrogens back in through ATP synthase. Okay, which is super weird to think about, but that's what happens. And then while this is happening, we're generating ATP, and guess what? That ATP is used to pump more sodiums out because remember how we have this issue now. There's too much sodium inside of the cell. So there's two ways that the bacteria get rid of sodium, here and here. Okay, those are the two arrows. And then there's three, I guess, regions where the sodium goes in. So it's still not perfectly balanced, but it's good enough. And in the process, the cell is also producing ATP and acquiring H plus. So good for this cell, figuring out how to survive in this kind of situation. Okay. All right, so that's for osmotic pressure. Now we're going to move on to oxygen, oxygen levels. So first, before we talk about different oxygen preferences, let's talk about why. Why do organisms even need oxygen? So you might remember this from Gen Bio, but during cellular respiration, organisms break down glucose to make ATP, and bacteria do the same thing. So bacteria are also performing cellular respiration. Now, in the last step of cellular respiration, if you remember, during the electron transport chain, the electrons are going through the chain, and then at the very end, they have to go somewhere. And it's almost always oxygen, which is what we call the final or terminal electron acceptor, right? So oxygen takes those electrons and then is converted into water. Oops, didn't mean to. Here, I'm just going to do that. Okay, so oxygen takes electrons, becomes water. It's the final electron acceptor. So that is why organisms need oxygen. But why not? Okay, so in this chapter, we're talking about preferences. Some bacteria like oxygen, some bacteria don't. So why do the ones that don't, why don't they like oxygen, right? Doesn't it seem like such a great molecule in this situation? But there are actually reasons why oxygen is quite toxic. And one reason is because in the presence of oxygen, you start to produce these molecules called reactive oxygen species, or ROS. These are very dangerous molecules because they are very, I guess, reactive, right? And what they do, or how they're produced, is when oxygen is present, it accidentally reacts with NADH dehydrogenase, which is a molecule or enzyme that is found in most bacteria, pretty much all bacteria. And then it also reacts with iron. So there's like a whole bunch of things that oxygen does. I'm sure you know that oxygen reacts with iron because you've seen iron rust, right? And how does iron rust? In the presence of oxygen. Anyway, so oxygen just reacts with a bunch of things inside of the bacterial cell, and it starts to produce these reactive oxygen species, which are super toxic. So some examples are a superoxide radical, which is this. Okay, so it's O2 minus with a little dot. I believe my OChem is really, it's been a while, but I think the dot is like an extra electron or something. So yeah. Okay, so that's one. Hydrogen peroxide, which is H2O2, obviously really bad. And then the hydroxyl radical, so that's the dot with an OH. So also super bad. So those are all, these molecules are accumulating when oxygen is present. So what some bacteria can do is produce enzymes to get rid of them. So for example, superoxide dismutase gets rid of the superoxide radical, and then it converts it into hydrogen peroxide. Now this hydrogen peroxide is also really bad, so it has to get removed. And so one way that it can be removed is through catalase or peroxidase. So a bunch of enzymes that can get rid of it. By the way, this H2O2, when it reacts with iron, like I said, gets converted into that hydroxyl radical. So that's how you create that. And so it's, again, very important that these bacteria produce superoxide dismutase and catalase so that they don't produce, because then it, it gets rid of this guy. And then when the H2O2 is made, it gets rid of the H2O2 immediately, which prevents this from ever being created. So all three of these enzymes are very important for bacteria to survive in the presence of oxygen. But some bacteria don't have these, right? Like some bacteria have these enzymes, some bacteria don't. So the bacteria that don't have these enzymes are not capable of living in oxygen. And so that's why there are some organisms that like oxygen, some that don't. Okay, so let's look at the growth preferences. So this is a test tube showing just like different oxygen levels, and they're just showing where an organism would grow if, for example, you had high oxygen, low, and no oxygen at the bottom. Okay, so aerobic organisms can only grow in oxygen, so you're going to typically see them growing at the very top of this test tube. Microaerophiles like oxygen, but only a little bit. They don't like too much. It's too much to handle. They just like a small bit. And so they're going to grow in this kind of low oxygen region. Then we have the anaerobes. The anaerobes absolutely cannot grow in oxygen. They will die. So those are probably the ones that don't have these enzymes, superoxide dismutase, catalase, or peroxidase. It probably doesn't have one or either or any of them. And so they will die in the presence of oxygen. So those anaerobes cannot grow in oxygen. Then we have two special guys, the facultative anaerobes. They will grow throughout the test tube. They can grow in oxygen and they can grow without oxygen, but they prefer oxygen. They're like, yeah, I would rather have oxygen as my final electron acceptor. But if it's not my final electron acceptor, fine, they'll figure something else out. So these guys can grow in both situations, but they do prefer oxygen. Whereas the aerotolerants, same thing, they can grow with or without the oxygen, but they don't really have a preference, so it doesn't really affect them one way or another. And so that's kind of the main difference is this has a preference and this one has no preference to whether it's living with or without oxygen. Okie dokie. Moving on to 5.5. Now we're talking about starvation. So obviously, sometimes bacteria have to deal with situations where there's not that many nutrients. So what really happens to the bacteria during starvation? So one thing is that they activate a lot of these starvation genes. And this is something we'll cover actually later. I think it's like chapter 10 or something. So don't worry about it right now, but they do have a lot of genes that help them cope with this situation. A lot of them change their growth pattern. So that's why I'm showing this picture. This is E. coli. E. coli doesn't look like that, but when it's starving, it does some weird stuff. And actually in the book, they said they don't know why. And I'm like, oh, great. Okay, anyway. And then the last one, very interesting, is that during starvation, they will kill themselves. So they go through programmed cell death, which, by the way, in cell biology, that's called apoptosis. Anyway, so these bacteria literally go through cell suicide to sacrifice themselves for the others. So they kill themselves, and the nutrients from that bacteria are basically given to other bacteria nearby. So it's like they're sacrificing themselves to be eaten by others, which is super weird. I didn't know they do that. Anyway, so let's talk about how that works, this programmed cell death. So there's a few genes. I know nobody likes genes, but we got to talk about some processes. So how does this programmed cell death work? So there's a couple genes here. So we have MASF. MASF is a toxin that these bacteria produce. And they produce it, and if this toxin is activated, it will kill this toxin will kill the bacteria. So it's kind of weird. The bacteria are producing something that could kill itself. Okay, but the bacteria also produce MAS-E. MAS-E is the antitoxin to MASF. Sorry, this should say antitoxin to MASF. Okay, cool. So MAS-E basically prevents MASF from killing the bacteria. So MASF is the toxin that would kill the bacteria. By the way, the bacteria are producing MASF, but the bacteria also produce an antitoxin against this toxin. So MAS-E prevents MASF from ever killing the bacteria. So that's what's happening here in part. No stress, I guess. So here's MASF being produced. Here's MAS-E being produced. MAS-E and MASF bind together, and so MASF never actually kills the cell. Okay? Again, both of these molecules being produced by the bacteria. Now, something that you want to note is that MAS-E gets degraded very quickly, but because the bacteria are constantly making MAS-E, then they're fine, they never die. Okay, but what happens during starvation is a change happens in this system, and so what happens is both MAS-E and MASF production stop. So here in starvation, the synthesis of MAS-E and MASF completely stop. Now, what ends up happening is Mas E is degraded very quickly. Mas F doesn't really get degraded, but Mas E is degraded almost immediately. So now that's bad, because now we have Mas F without the Mas E. Remember, Mas E is the antitoxin and Mas F is the toxin. If Mas E is gone, but Mas F is still present, then the cell ends up dying. So Mas F, which is still present, causes mRNA to be cleaved, causes the growth to stop, and then ultimately the cell dies and it kills itself. Okay, so that's how the trigger happens. And so again, Mas F causes the cell death. So that's all cool, right? Good for this bacteria, just sacrifice itself for its neighbor. But then the next part is really what I think is fucked up. So what the cell also does while it's dying is it gets a G6PD. So G6PD stands for glucose 6-phosphate dehydrogenase. But you can just put G6PD, but yeah, I could just write it down here. So it's glucose 6-phosphate dehydrogenase. Okay, anyway, G6PD is an enzyme. And it gets degraded. So these bacteria produce a protease that degrades this enzyme into these tiny little protein segments, right? So G6PD is a giant protein, and this protease breaks it up into tiny little pieces. Okay, what the hell? And this pieces of G6PD are going to travel to a neighboring cell. Okay, not this cell. Okay, this has nothing to do with this cell. It actually travels to a neighboring cell. And what it does to the neighboring cell is that it inactivates, guess what? Mas E. It inactivates the Mas E of a neighboring cell. Well, that's screwed up, because remember, Mas E is the antitoxin. And if Mas E is inactivated, then now Mas F is free to kill this neighboring cell. So the neighboring cell has active Mas F. Remember, Mas E is now occupado. And so Mas F is active, causes mRNA cleavage, causes growth stasis, and then eventually causes cell death. So this whole beginning, so, you know, up until here, all we were talking about was how the cell kills itself to sacrifice itself for its neighbors. But then it also kills its neighbors. It degrades G6PD, which inactivates Mas E nearby, and it causes the nearby cells to also die. So it's like, wow. So yeah, it's a 2-for-1. One of the bacteria die, and it takes its friend with him, basically, which is quite interesting. But this is all, again, for sacrificing themselves for, I guess, the younger cells or whatever. But it's funny to think about, like, okay, but what if all the bacteria decide to kill themselves? Then everybody's just dead. So I always, the book doesn't really explain, like, who decides to kill themselves and who decides not to die. So yeah, but this is how it works. So okay, last part of the chapter, we're going to talk about how we control microbes. So human beings are obsessed with controlling microbes, preventing them from growing, making them grow, whatever. But in this case, we're going to talk about preventing the growth of microorganisms, which obviously is super helpful when we're trying to, you know, cook food, right? We don't want our food to be contaminated. We don't want our materials that we use in the lab to be contaminated. So there's a lot of reasons why we want to control microbes. So a couple definitions. Remember, sterilization is killing all living cells. That includes killing spores and viruses as well. Okay, viruses are destroyed in this process. Whereas disinfection, remember, is killing and removing disease-causing organisms. So we're not killing everyone, we're just deliberately killing the ones that would cause disease. Okay. An antiseptic is the same thing as disinfection, except specifically for living tissues. So we're disinfecting the skin, for example. So when you, you know, put alcohol, when the nurse puts alcohol on your skin before they give you like an injection or a shot, that is antisepsis, right? They are trying to disinfect your skin. So yeah. And then finally, the last one, sanitation. We're trying to remove the microbial levels to get it to a safe level. So we know that we're not killing everything, but we're at least decreasing it enough so that it's not filthy, is essentially what we're doing there. Okay, so there's a couple ways that we can sterilize things, and one of them is through the autoclave, which we talked about in the lab. So autoclave sterilization. Why do we do autoclave sterilization? Why can't we just boil things, right? Well, it's all about the spore. So remember we talked about how endospores are these dormant states that some bacteria have during very extreme environments. Well, one of those extreme environments is extreme temperature. So endospores can actually survive in very high temperatures, including boiling temperatures. So if you were to get something and just boil it, and there were endospores, you would probably not kill all of those endospores, which is obviously not good. And so the autoclave is the best way to kind of alleviate this because we can reach 121 degrees Celsius and the pressure is 15 psi. And so you can do this for 20 minutes. Now, why does this work? Why does the autoclave work? Well, increases in pressure cause an increase in the boiling point. And I'm not going to go over the chemistry of it, so you're just going to have to review your general chemistry, but there's like a relationship between pressure and boiling point. And so when you increase pressure, you increase the boiling point or the boiling temperature of water. And so what this does is it allows us to bring your sample to 121 degrees Celsius, which is way past boiling, but the actual water doesn't boil. But the spores totally get killed in this situation because they've never experienced something that high, right? Think about a bacteria. Most bacteria live in, sorry, there's something on my screen. Okay. Most bacteria live in liquid situations, right? So even bacteria that live in the hottest liquid temperatures are living at maximum boiling temperature because past boiling temperature, there's no water, right? Water is now steam or water has now become water vapor. So when you think about the normal conditions that a bacteria could ever experience, boiling temperature is typically the maximum. You don't really think of bacteria living past boiling temperature because then they would be living in gas, right? That wouldn't make any sense, or steam. So we bring these bacteria to 121 degrees Celsius, which is something they've never experienced before. They're dead, so the spores don't survive that. Okay. Another way to, not necessarily sterilize, but usually we just consider it disinfection because it doesn't really kill 100%. But anyway, one way that we can get rid of microbes is through pasteurization. So there's 3 types. There's low temp, long time, which is LTLT. Guess what? Low temperature, so 67 Celsius for a long time, 30 minutes. High temp, short time, by the way, is also called flash pasteurization, which is a common way that we have pasteurized milk. So 72 degrees Celsius for 15 seconds, which is pretty short, right? That's like a significantly different time than 30 minutes, right? Imagine if you had milk at 63 degrees Celsius for 30 minutes. Remember, 63 degrees Celsius is like almost double body temperature. And you're keeping milk at that temperature for 30 minutes. So that sounds like it's disgusting. But, wouldn't the milk curdle? But when you have the milk at 15 seconds for 72 degrees Celsius, that's like way better, right? And then we even invented ultra temp or ultra high temp pasteurization, and that's at 134 for 1 to 2 seconds. So it's just like flash, not even flash pasteurization, instant pasteurization. So 1 to 2 seconds for really hot temperature. Okay, another way that we can get rid of microbes is through filtration. So again, we also talked about this in the lab. So we can put liquid through a filter, usually 0.22 micrometers, and those pores of the filter are so small that even the bacteria cannot pass through. And so when you push the liquid through, the liquid that comes out is sterile because there's no bacteria. Okay, and then of course we can do this with air, so through HEPA filters. So HEPA stands for high efficiency particulate air, and so these HEPA filters are pretty much the same as the 0.22 micrometer filters, except they're just 0.3. But anyway, they remove the microbes from the air by filtering them out. Okay, and then the last thing I'll talk about in this chapter is disinfectants or chemical control of microorganisms. So they have like a little chart about the different types of disinfectants. So phenols, alcohols, aldehydes, all of these pretty much target lipids or proteins or DNA. They have some kind of target. Primarily it's lipids and proteins, but also DNA is another target. So those are really, obviously helpful for getting rid of bacteria. Then there's the quaternary ammonium compounds. So they're here in the 4th part of this table. And these guys are positively charged ions, as you can see. And when they come into contact with the bacteria, they actually disrupt the cell membrane. The cell membrane becomes like almost completely wiggly, and then the bacteria explodes. So really effective way of getting rid of these guys. And then finally, there's gases. So we talked about this in the lab. Ethylene oxide was one of them. They usually disrupt proteins, yeah, in the cell. So, yeah. All right. And then the last thing we're going to talk about is antibiotics and biological control. So we're not going to talk about this in too much detail. Antibiotics are these compounds produced by microbes, and they are able to kill or inhibit other microbes. So that's kind of the definition of an antibiotic. I'm not really going to cover this too much because we literally have a whole chapter on antibiotics, so that's all I'm going to say. But the other thing we're going to talk about is biological control. So biological control means that we are using something that's living to control something else that is living. So for example, if I have, an issue with salmonella, like, oh, there's so much salmonella here, I got to get rid of it. Instead of using antibiotics or antiseptics, you can use another living thing to get rid of the salmonella. So an example of biological control that you might have heard of was like in Gen Bio is like during farming. Maybe you like learned this in like not really evolution, environmental science. So for example, when there's like pests, like these little like bugs that eat the crops in farming, what they do is they like bring in, I don't know, other bugs like ladybugs to eat the bugs that are causing the problem. So that's called biological control. You're using one living organism to get rid of the other living organism that is causing your issue. So yeah, one way that we can do that is through probiotics. So when you consume probiotics, you're taking in gut microbes that are good for you. And the hope is that they will outcompete the bad microbes that are already in your gut. So that's one way. Another way is through phage therapy, which, by the way, I think is kind of weird because phage therapy uses bacteriophages. Bacteriophages are these viruses that infect bacteria. So that's all you need to know for now. So a virus that infects bacteria specifically. And we use these viruses to specifically kill the bacteria that's causing a problem. So for example, if we discovered a bacteriophage that kills salmonella, instead of giving a person who has salmonella poisoning antibiotics, we can give them a phage, and that phage would go into their stomach and infect all of the salmonella, and then the salmonella are gone, right? So that's a really cool new, I guess, way of fighting bacteria or getting rid of bacteria is by killing them with their worst enemy, which are the phages. But the funny thing is the word biological control implies that we're using something living to control something else that is living. Now with the phage, since it's a virus, it's not technically alive. So I just think that's funny that we use that term, biological control, for phage therapy, even though it's not really alive, but whatever. Okay. Anyway, that's pretty much it. So thanks for watching, and I will see you in the next video.